Good morning. It's October 23rd, and this is your daily brief in science. Here's everything you need to know. Recent research calls for a shift in how antidepressants are prescribed, emphasizing personalized care, routine health checks, and shared decision-making. The study suggests that treatment should be guided by individual health profiles and patient priorities, such as avoiding weight gain or blood pressure increases. For example, some medications like amitriptyline are linked to weight gain and increased heart rate, while others like sertraline may lead to weight loss and lower heart rate. The research analyzed over 150 studies involving more than 58,000 participants and found notable differences in side effects among various antidepressants. Certain drugs, including nortriptyline and doxepin, can significantly raise blood pressure, raising concerns about cardiovascular health. In the UK, prescribing practices currently favor a small group of SSRIs, such as citalopram, sertraline, and fluoxetine, which tend to have fewer physical side effects. To support personalized treatment, developers are creating a free online tool for clinicians and patients to compare side effect profiles, though experts note that long-term effects still need further study. Health organizations emphasize the importance of routine physical health monitoring and informed discussions, especially since short-term physiological changes can accumulate over time. Advocates in mental health are urging a cultural shift in the UK, where 85% of prescriptions are for just 3 antidepressants, to promote more individualized approaches. Clinicians highlight that side effects can vary based on factors like metabolism, age, and other medications, underscoring the need for careful monitoring. Experts warn that findings from short-term studies may not fully reflect long-term or real-world scenarios involving older patients or those with comorbidities, indicating that further long-term research is necessary. Researchers have replicated the zebrafish's optomotor response using both simulations and physical robots, demonstrating how these fish maintain position in flowing water. This work helps to reverse engineer the neural circuits involved in this behavior. The study shows that vision alone can be sufficient for zebrafish to perform certain actions, challenging previous assumptions that multiple sensory inputs are necessary. Additionally, scientists identified 2 previously unknown neuron types that play a role in visual response behaviors. This discovery deepens our understanding of the neural mechanisms in zebrafish. The research emphasizes the importance of integrating neural, physical, and environmental factors to understand vertebrate behavior. It also suggests that multisensory feedback could enable injured vertebrates, such as eels, to continue swimming after spinal injuries. These findings provide insights into evolutionary adaptations from aquatic to terrestrial environments. The open-source simulation and robotic tools developed by EPFL are designed to facilitate further research into visuomotor coordination across species. Overall, this body of work highlights the value of combining neural studies with embodied models to better understand behavior. Neutrino experiments are shedding new light on some of the universe's biggest mysteries. 2 major projects, NOVA in the United States and T2K in Japan, have teamed up to improve our understanding of neutrino behavior and their possible role in explaining why matter dominates over antimatter. These experiments focus on measuring neutrino oscillations with unprecedented precision to see if neutrinos and antineutrinos behave differently, a key factor in this cosmic puzzle. A central unresolved question is the mass ordering of neutrinos, specifically which neutrino is the lightest, though recent studies have narrowed down the differences in their masses. Both NOVA and T2K use long baseline setups, sending beams through Earth's crust over hundreds of kilometers. NOVA from Fermilab to Minnesota, and T2K from Tokai to the Super Kamiokande detector in Japan, to observe how neutrino flavors change during their journey. They utilize advanced detectors to analyze these oscillations, producing high-precision data that show their findings are compatible despite different experimental setups. This collaboration highlights the importance of precise measurements in advancing fundamental physics. Future experiments like Fermilab's DUNE, Japan's Hyper Kamiokande, and China's Juno are planned to further explore neutrino properties. DUNE, in particular, aims to definitively determine the neutrino mass ordering. These upcoming projects are expected to deepen our understanding of neutrino behavior, with potential implications for the universe's composition and the fundamental laws of physics. Neutrinos are extremely lightweight particles that can change flavors, electron, muon, tau, as they travel, a process called neutrino oscillation, which depends on their mass states. They originate from cosmic events like the sun's core and supernovae, passing through matter undetected. Studying neutrinos could help unlock mysteries related to the matter-antimatter imbalance, dark matter, and dark energy. Recent research has identified the gene HMGN1 as a key contributor to congenital heart defects in individuals with Down syndrome. Down syndrome is caused by an extra copy of chromosome 21, which leads to multiple genetic changes. Experiments with human stem cells and mouse models show that overexpression of HMGN1 disrupts normal heart development by reprogramming cardiac cells. This disruption results in malformations associated with congenital heart defects. Scientists suggest that therapies aimed at reducing HMGN1 activity during pregnancy could potentially prevent these defects. The research also explores how HMGN1 interacts with other genes like DYRK1, influencing heart development through complex genetic interactions. The study highlights that combining genomic and computational methods can help understand other chromosome-related disorders, such as intellectual disabilities. The genetics of trisomy 21 involve multiple loci, including DYRK1A and interferon genes, which contribute to the risk of congenital heart defects through multigenic and epigenetic effects. Led by Sanjeev S. Ranade, the team demonstrated HMGN1's role by deleting one allele in trisomic cells, which reversed abnormal gene expression. HMGN1 affects chromatin structure and gene regulation, impacting key cardiac genes like TBX2 and TBX20. This research underscores the potential of integrating genomics, AI, and animal models to develop targeted therapies for complex congenital disorders. While therapies such as pleiotrophin show promise, their application in humans is still distant. Overall, these findings advance understanding of the genetic and epigenetic factors in Down syndrome and open new pathways for intervention. Researchers have used CRISPR gene editing to develop pigs resistant to classical swine fever by targeting the DNA JC14 gene, which is crucial for the replication of pestiviruses like CSF. In experiments, these genetically modified pigs showed no symptoms or detectable virus in their blood, unlike control pigs that displayed typical signs of the disease. This is the first demonstration of resistance to CSF through gene editing, offering a potential alternative to traditional vaccines and large-scale culling during outbreaks. The research team emphasizes that ethical, legal, and safety issues must be addressed before such technology can be used commercially in livestock. Future studies aim to determine if this resistance trait can be inherited across generations, which would eliminate the need for repeated treatments or vaccinations. Classical swine fever continues to pose a significant threat, with outbreaks historically leading to extensive culling, such as in the Netherlands in the 1990s, and ongoing issues in countries like China, Russia, and Brazil. Current control strategies rely heavily on live virus vaccines, which are labor-intensive and restrict trade, and pose risks if vaccination efforts are interrupted. Preliminary observations show no adverse effects in the resistant pigs, but further research is needed to assess long-term safety and animal welfare, especially since traditional breeding does not typically prioritize these factors. The same gene involved in CSF virus replication could potentially be targeted in other livestock, including cattle and sheep, to develop resistance against related pestiviruses like bovine viral diarrhea and border disease. Researchers are exploring whether similar gene edits could confer resistance in these animals to improve health and productivity. Regulatory barriers currently prevent the sale of gene-edited pork in the European Union, though the United States has approved such animals, including pigs resistant to porcine reproductive and respiratory syndrome in 2025. The resistance provided by the DNA JC14 gene modification appears stable across species, but long-term studies are necessary to confirm its durability and safety. This breakthrough represents a significant step forward in combating CSF, with potential to reduce disease impact and lessen dependence on costly, labor-intensive vaccines. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.